Hi everyone, welcome back to another live stream, live tissue portal live stream. Please let me know, whoever joins, let me know if you can hear me. I may have to mute my computer. There we go. So today we're doing some bulking. So I'm bulking some plants. So I need more plants of more containers. So I have this Philodendron caramel marble. I just did Philodendron Spirit Santi. Yeah, so I just did Philodendron Spirit Santi. I was like, yeah, why not just live stream it? So. Want to check the audio pretty quick. Take a look. Oh, okay, I can hear me. Okay, good. Okay, so I already have cleaned my area. Hey, we have some viewers now. Okay, so I was saying I'm working on bulking some plants. I just need to have more of everything. So today I'm working with a Philodendron caramel marble. I just did a Philodendron Spirit Santi, and but I said like, well, why not just a live stream as I'm here? So I want to spray it with alcohol. Let's open this up. This container has so many plants. So I'm just going to take everything out and start splitting them. So I'm going to open half. Then I take the smaller one. So I want to do this like super fast. This is just a quick bulking. I'm not really care that much about removing all the roots or everything. I just need to do as many as I can pretty fast. I'm also doing this as a test for the live streaming, as I had to change my settings on my computer, and just testing out to see how it works. So later on, after we're done with the live stream, I want to check it out to check the video and audio, the settings, see if I need to change anything else. Anybody on the chat can tell me, like, you can hear me okay, if it sounds okay, or if the sound is bad. I'm under the lamina flow hood right now, so the audio won't be great because I mean, it's picking up the audio from the hood, from all the air. But okay, so there we go. So I just made split them as much as I can, and just like a salad. many plants, probably over at least 100, definitely more than 100. But like I said, I just trying to do this as fast as possible because I have so many plants to get through. So let's sterilize my tooth one more time. I'm going to spray my containers, more deli cups. Alright, let's start moving plants in. This is going to be super fast. So I'm going to take like 3 smaller clumps like that. And this weekend is the masterclass, the online tissue culture masterclass. So you're interested on learning more about tissue culture in just 2 days. Um, you can check the link in the description to register for the class. It's this weekend, so this Saturday and Sunday, and so you only have a couple of days to register. I already have my plan set, I have my media ready, I have I just need have my media ready, I have my plans ready, I'm ready, so ready for the next class. Exciting, actually. I need to double check how many students we had over the master classes, but it's very incredible to think that I have a teach over 100 people or students that I have during the classes. So the tissue culture I'm doing right now, you can see I'm doing this like super fast. You will not see this in any other social media. Any other social media, you will see like doing like a really neat and pretty job. You will definitely not get that right here. I just doing this super fast. Still keeping everything sterile. I still need to be very careful and make sure not to contaminate my cultures. But I'm not really. I don't really taking care of all the roots or the splitting every single plant. Just working by efficiency because I have so many other plants that I need to move. I made 20 liters of media, so I have a lot of media to get to. This is Philodendron caramel marble. So super fast. This is very easy, and essentially I'm doing like from one container, from 1 cup, I'm going to 20 cups, and I'm doing this. We'll see how long it takes me, but so far it has been like less than 10 minutes. I'm already on almost 15. So this is a process that is very fast. and also like if I'm putting like close to like 5 to 10 plants on each cup, in 3 months they will be full. Like you go back to where when I got the plants out of the container that I'm working now, it's, they have over 100 plants each. So you can see that I can go from these 20 containers in about 3 to 6 months. I will have 100 plants on each container. Split some of these plants. And I see there are 19 viewers, so if anybody has any questions about tissue culture, feel free to ask. It's easy for me to keep the stream because talking by myself is not easy. So if anybody have any questions about tissue culture, just split these plants a little bit more. And there we go. So I'm done with the caramel marble. That was super fast. It took me like, what time is it? It took me like 10 minutes to do. So that was pretty fast. I'm not going to wrap them yet. I will do that after I'm done with everything. So I want to move this. I want to bring more. So I'll be right back. Alright, so I'm back. Have another plant. This is a Philodendron giganteum. I will clean my plate first. What plant are you working out with? I just finished Philodendron caramel marble, and earlier out of camera, out of the live stream, before I start live streaming, I was doing Philodendron Spirit Santi. I'm just bulking plants right now because I need to have more plants, and I was running low, so I just doing a lot of them. Like a lot. I made 20 liters of media, and I did a lot of carnivorous plants as well. That was on Monday. I did 20 liters of. I did 100 carnivorous plants, 100 containers of the carnivorous plants. I need to get back to the carnivorous plants. Well, I'm using egg instead of biocoppers. Right now I'm just bulking. I need numbers. I need numbers of containers. I need to have as many containers as I can at the moment. So I'm just trying to get as many containers as I can. So with the biocouplers, I will get a lot more plants, but I don't have the the biocouplers at the moment. I do have a bunch of biocouplers, but they are in use. And on inventory that we had to keep up with the inventory. I cannot just grab all the biocouplers that we have here in the stock because we also sell them, and we have to make sure we have enough for to sell. So spraying everything. Okay, so let's start by moving this other plant. So again, this is Philodendron giganteum. I want to split it. I want to do this super fast. So this is something that you don't see on social media, where they show you like how to do this like super neat and super nice. Not here. I want to do it like super fast, and it's still going to be clean. Still working on sterile conditions and trying to keep everything as stay clean. So, how long do you store the oxygen cytokinins? So depends on which ones, but in general rule, you want to store them. Well, they have a for the solutions, for the liquids. They have a 6-month shelf life, and for the powders, they have usually 3-year shelf life for powders. For the solutions, you want to keep them in the fridge. For all of them, except for there are a couple that you need to keep them in the freezer. So whenever you on the website, we have the instructions on how to keep them. That's something that you don't have. It's something that is public knowledge, or it's something that we keep on the website. That the storage temperature and how long it's stored, you can keep it for. Now I will tell you that we make plant regulators, the stocks, to sell almost every day, because we make this as fresh as possible. As we get the orders, we pretty much make them. Make the plant regulator solutions, because like I say, they have a 6-month shelf life. So we want them, whenever we sell those to you, you get this pretty much as fresh as possible. So we only make, we don't make a lot at the time. We just make whatever we are selling pretty much. So that way we keep it, you know, you have a fresh solutions. So I'm just going to remove all of this dead plant material. Like I said, I'm just doing this as super fast. Let me check another question. What's a good means to create callus? So I don't know what people are so intrigued about growing callus. You don't want to grow callus. If you're not doing any molecular genetics, if you are not doing plant transformation, don't bother with callus. You don't want to grow callus. That's my opinion, but because when you grow callus, I mean, those are undifferentiated cells. So that means that you can create different mutations where, oh yes, it can be desirable, but if you have already a plant that is valuable because of the traits that it has, it's better just to keep it as close to the mother plant. You just keep it as an identical clone to the mother plant. You don't want to do any like modifications to it. So I just stay away from callus as much as possible. Now you want to grow callus, I mean, I can tell you, you just need for the most part, NIA, TDC, 2,4-D. Those are kind of the hormones that you will use to grow callus. Well, the concentration, they kind of depends on the plants that you're working with. All right, let's see. These plants, what plant regulators are in? I mean, you see there are a ton of plants, and so many. So they were growing in VIP. The concentration, I had to look on the date, so I can look on my notes. But usually I cannot. Well, I usually do between 1 and 3 milliliters per liter, and they get a good, very great, good amount of multiplication, as you can see. So um, 2 milligrams of 2,4-D and 1 milligram of kinetic for callus in majority of plant. Yeah, that's about right. We found like NAA, usually don't for callus, well, yeah, kinetic, but probably VIP, I found that probably it's a little, you get a little bit more callus, or TDC, no, I'm sorry, TDC is the one that you need for callus as your cytokinin. So there we go. So you see all the plants that I got. I have a bunch of trash. Now all this trash is actually like rhizomes, so I can actually plant this and get more plants, but I just wanted to get the plants. I don't want to bother with that right now. Sterilize my tools for a moment. Take all of this out. Callus is good for suspension culture. Yes, but like I say, you are doing. I mean, I guess you're probably working on a lab, or like for transformation, or that's probably why you want callus. But other than that, you don't really want callus. I hear that they grow coffee callus culture for consumption. Had to look into that. Alright, so I have my plants, have my containers. I'm going to spray those with alcohol. Let me cool down my tools for a moment. So let's get the plants in. Oh, okay. So yeah, that's why I work with a plant cell suspension. Yeah, that's why. Yeah. Oh yeah, but you were not the person who asked about the callus. That was another person. Where's my? Who asked for callus first? Oh, I cannot find who was working with us. Oh, yeah. A session garden was asking for callus. All right, let's put some plants in. So I'm just going to put like 2 plants in there, because I'm not going to spread them like super nice. Mostly because I don't have the time to do it because I have so many other plans to work with. Yeah, so today we are only streaming here on YouTube. I had to, like I say, I'm working on different settings on my computer right now, so I need to test the other live streams as well. So we have only YouTube today. Maybe next week we could potentially live stream also on TikTok and Instagram as well. Can I put use media to compost? I probably not. It's probably going to grow mold. You're trying to compost your old media. Just a reminder that this weekend is the upcoming Tissue Culture Masterclass, the online version, so anybody in the world can join the class. We are also doing a class giveaway, so check our Instagram page if you want to you can enter to get the opportunity to get a free Tissue Culture Masterclass. Just make sure to check our Instagram post on how to do that. It's like a plant tissue culture, like a scholarship, where you can take the class with me for free. Oh, by the way, I don't know if you've seen our latest video on the Nepenthes tissue culture that me and Lauren Plants in Jar film. I actually have an update, so the Nepenthes are actually growing. I will have I can show you here in a moment. But if you haven't seen the video, you can check it out after the live stream. But me and Lauren, we record a video on how to sterilize Nepenthes. So you can introduce them to tissue culture, and they have started to grow. They are growing now. So I will show you. I will show you the cultures here after I finish with these plants. You're having trouble to get the plants to get the gel, your media to gel. Let me see. What was it? All right. I was wondering, does the pH have to be on point when autoclaving media? Usually, I see that the pH may not be the biggest thing. It may be so you can tell me, is some of your media, this is what I see the most, is some of your media like not gelling at all, like really liquid, but some are like super solid, because that's what I see a lot. if some of your media is really liquid and some is really solid, you need to make sure to mix it up. Make sure to it's mixed properly mixed when you're pouring the media. I also made a video on how on the easiest way to make a tissue culture media. That was from you can check on the channel. That video I show you how to make tissue culture the easiest way, like by far the easiest way to make tissue culture media. So you can check that out. There we go. So this is the last one. All right, so I'm done. I have the 20 right now. Let's see. Yes, still water. Okay. Yeah, so that's what I see a lot, that when the media is like very liquid or very solid, it's mostly because you haven't mixed it. You need to, when you're pouring, make sure you are mixing the media. Because what I see a lot of the times is where they have like the media, they make the media in the pitcher, and then they pour and pour and pour and pour. And what they do in that, all the media, all the gel, all the agar or gel and gum is just settling down in the bottom of the pitcher. So when you're pouring, the first one, you are pretty much pouring nothing, no agar or gel and gum. And the very last ones, they have all the agar there because everything has settled down to the bottom. So need to make sure to, when you pour, agitate, pour, agitate, pour, agitate, pour. However, like I say, just check the latest video. Well, not the latest video, but the video on the easiest way to make tissue culture media. I show you a little trick using like a spoon where you add the gelling agent directly to the containers instead on the entire pitcher. So before you're pouring it, so you add that after you're pouring it. So let me go grab the Nepenthes that me and Lauren did, plants in jars, so I can show you like a little update. So I'll be right back. All right, so I'm right back. So these are the Nepenthes that me plants in jar did for the video. Let me show you. So let me change camera again. So this is the first one. It is growing a little bit of bacteria. It's not a lot, but it's a little bit of bacteria that I see right here. So I will take this out, not today. I'll do it during the class. So you're taking the class, great. If not, I mean, you can go subscribe to the class, to the online tissue culture masterclass. The link is in the description. Actually, 1 second. I'm done with the tissue culture today, so I can take the turn off the laminar flow hood. Well, yeah. So for class, I will do this in class. I will re-sterilize them because they are growing a little bit of bacteria. You can see that better right there. So a little bit of bacteria growing in there, but it's super hard to see. Let me see if I can get even closer. It's not going to focus. That little point. Let me see. There is a little green point right here. That's the meristem, and I can see that it's growing very slowly, but it's growing. It's getting there. So I will move this I'm going to re-sterilize it and place it in a test tube with fresh media. But I will do this in class. The second one, let me show you this other one first. So this is the another one that we did. And this one also has a little bit of bacteria. So like I say, Nepenthes or carnivorous plants, they are some of the most difficult plants to sterilize. They are so difficult. That's what we did. If you saw the video, you saw that we used sodium dichloro for 24 hours. We did at 250 ppms. And you see, I mean, we even with that, we still got a little bit of bacteria, but it's not much. So I can do a re-sterilization on this one. You see right there, but the plant is growing. It's definitely see that the meristem is starting to grow in there. And the last one, this is the one that I'm most excited about, because you see that little point right there. The little dot right there is a new meristem. So this is actually Lawrence. This is the one that she did, and you saw on the video, she cut it like super close to the meristem, but just there, like you see that it is growing, the little point. So it has been like the 8th, today is the 20th, so it has been just 12 days, and it's starting to grow. I mean, the Nepenthes is very slow plant. It does have a little bit of bacteria as well on this side. You see it right there, a little bit of bacteria. But like I said, I will sterilize this in class. So I'll show the students how to re-sterilize these tissue culture plants. I need to grant this stuff out here. So, I made like a bunch of cultures today. I have a lot more to do, but I'm not going to do this in camera just because it kind of slows me down a little bit. But I just wanted to kind of come live again, because it's been a couple of weeks since I have done a live stream. So it's great. Hopefully next week I will go live as well on Instagram and YouTube, and TikTok. I just kind of changed some of the settings on the camera and the microphone. I'm using now this microphone right here instead of my regular microphone, so hopefully the audio is better. Now obviously I was working in the laminar flow hood, which makes it pretty loud, but hopefully now, because the microphone is closer to me, you can hear a little better. And but yeah, the class is this weekend, so you have this Saturday and Sunday, so you have only a couple days. You have a couple days to register for the class, and everything that I have used in this live stream, we have all the containers, the snap up containers, the test tubes or media, plant grow regulators, or the biocouplers. Everything is available on plantcelltechnology.com. I think I'm going to. Oh, let's see. Another questions. Appreciate your lives. Thank you. Yeah. Oh, yeah. For the media, make sure to, whenever you are pouring, make sure that everything is in solution. Don't let it settle down to the bottom, because otherwise it's going to happen that some of the media will be like super solid and some will be like liquid, because it's not going to be solid. It's not like properly mixed. So I think that's everything for today. I'll come back next week on a live stream, and also have a couple of more videos coming up pretty soon. I'm very excited. So I will see you. I'll see you hopefully in class. So make sure to register for the class. All right. Goodbye. And oh, okay. Let's end right there.